Yeah. But you're excited getting that.
Should we get started with the lone 5 in person? We have 9 online, so 14 strong today. Woo! All right, awesome. Well, we'll start our micro discussion today. I have a lot of resources online in the folder. We're going to start with kind of the intro micro lecture because it goes through a lot of the tests that you'll need to know for NPLEX in regards to microbiology. It also goes through different virulence factors, things that are not related directly to bugs and path. So we're going to go through this lecture first, and then we'll get into some of the most important bacteria that you should know. Oh, hey, good morning, peeps. Most important bacteria you should know. We'll touch on viruses today. I posted a few things on parasites online and fungi. We probably won't cover those in class because at this point, it is not high yield for you to go over fungi or parasites. Really, bacteria would be your top at this point, and then viruses if you're feeling comfortable about your bacteria. That's what I would lean into at this point in the process. So that's kind of what we'll reflect on today. So this first one, it's the the 1 underscore microbiology presentation we'll go through first. So first, just basics, a few structure of bacteria components that is important to know. These things sometimes do get tested on NPLEX, so that's why they're included. Your appendages for your bacteria, your flagellum or your pili or fimbria. So flagellum is really going to be your motility. So if a bacteria is motile, you can assume that they may have flagella of some sort. And so sometimes you could see NPLEX asking those questions about motility or bacteria motile, and they may use motility or flagellum or flagella interchangeably sometimes on NPLEX. There's typically, so if that is the case, and then you could assume your bacteria is motile. Pili or fimbria, these are glycoproteins. They help bacteria adhere to cell surfaces, or they're helping with reproduction of bacteria during conjugation, transfer of DNA from one bacterium to another, which is how bacteria can actually gain DNA from each other. They can mutate or they can develop into have new characteristics. Oftentimes different toxoids are shared between bacteria. So these would be the only 2 appendages I would care to know about. Then we get into the outer structures of the bacteria. These are where it becomes a little bit more important. Capsules, not all bacteria contain a capsule. They are polysaccharide in nature, and they protect against phagocytosis. Bacteria that contain capsules are unable to be tagged initially or identified initially by our macrophages and digested and presented. So they have to be opsonized via antibodies. And so that process takes longer. So we'll look at some encapsulated bacteria, but this is a virulence factor that can make the disease more severe because the bacteria will take longer to be identified and can hang out in the body and grow and develop further before the immune system essentially eliminates it. A slime layer is more of a looser network of polysaccharides. So these are both polysaccharide layers, but slime layer is less organized of a layer around the bacterium. It is more involved with cell adhesion, but it also supports creation of biofilms. So a bacteria that forms slime or has a slime layer can form biofilms. Not all encapsulated bacteria can form biofilms. Most bacteria that have a slime layer can form a biofilm. We see biofilm formation, especially in our gut bacteria or in bacteria that are indwelling in the GU, genitourinary tract, indwelling catheters, things of that nature. Obviously, biofilm formation from bacteria is going to be much harder to get rid of because these bacteria have essentially created a protective bubble around themselves, almost like what an abscess is. They're protecting themselves from identification, from tagging via antibodies, and from phagocytosis. So our encapsulated bacteria, let me see if I can move this. There. Our encapsulated bacteria, you should know, is please shine my skis. I mentioned this, I think, yesterday, but Pseudomonas, Streptococcus pneumoniae, H. flu, Neisseria meningitidis, E. coli, Salmonella, Klebsiella, and Group B strep. These are your important encapsulated bacteria to know, meaning that they have to be opsonized to be able to be digested or be able to be identified. 3 of these bacteria, Strep pneumoniae, H. flu, and Neisseria meningitidis, are unable to even be opsonized. So these bacteria are ones that we've actually looked into and we've created vaccines for. And so there are vaccinations for all these 3 bacteria because they're exceptionally hard for the immune system to clear. Now, they're not impossible. They're not always deadly. Strep pneumoniae, common cause of pneumonia, we can treat that with antibiotics. But H. flu and meningitidis, these are causes of meningitis, and they can be dangerous potentially to especially infants or children, I mean, really anyone, but especially infants and children. So we have vaccines to help protect against these 2 species. Oops, there we go. What did my thing die? Oh, so sad. Okay, that's fine. All right, then moving into other things that surround our bacteria that are important to know is our cell envelope or cell layer. So the outer membrane. This is where we get into discussion of gram negativity and gram positivity with our cells or with our bacteria. So the outer leaflet layer can contain endotoxins, LPS or LOS as examples. They have lipids on them, so that's the L in the LPS or LOS. Lipid A in particular induces both tumor necrosis factor, alpha, and IL-1 in the immune system. So when the immune system recognizes that an endotoxin is present, tumor necrosis factor alpha and IL-1 in the immune system will be secreted by the cells that identify that. And then that will increase an inflammatory response, cause a fever, cause inflammation. It's very pro-inflammatory. That's why bacteria that contain endotoxins typically cause a lot more symptoms than bacteria that don't, because the body is creating this huge pro-inflammatory response when it identifies this. There are also other embedded proteins in the cell wall and the outer membrane, just like any cell that we could see. But these cells, these bacteria, they have porins, which can transport things across. And then their inner leaflet is a phospholipid bilayer. So it's very consistent with what we would see. Gram-negative bacteria only are the only bacteria that have this outer membrane layer with endotoxin present. Okay. So gram-negative bacterial cell wall. When we talk about some other components of the cell wall, we can see here this outer membrane, a peptidoglycan cell wall, and an inner membrane. This is gram-negative bacteria. So outer, this small layer of peptidoglycan and inner, versus our gram-positive bacteria has this large layer of peptidoglycan and then only an inner membrane. It does not contain an outer membrane. And so this will come into play when we talk about staining. The gram-positive bacteria do not have an outer membrane. They have a larger peptidoglycan cell wall. Gram-negative bacteria have 2 membranes, an outer and an inner, and they have much less peptidoglycan. So this is why bacteria stain differently. We'll look at those stains here in a moment. But some definitions: the periplasm is essentially the space between the cytoplasmic membrane and the outer membrane, so between these 2 spaces here. You can have substances that can accumulate in this space as they exit to bacteria, enzymes in particular, and this is only present in gram-negative bacteria, as I mentioned. Peptidoglycan, a thin layer, is also present. The cell wall is made up of peptidoglycan, a protein sugar backbone. It has these peptide side chains. They're cross-linked, so they're very hard to break down. It's a protective structure for bacteria. They provide rigid support, and they really protect against osmotic damage. So that's why bacteria are not as affected based off of the environment that they're in regards to osmosis, because they have this cell wall that's preventing water from flowing in or water from flowing out. We see a larger peptidoglycan cell wall in gram-positive bacteria and a smaller in gram-negative. And then on the innermost membrane, we have a cell envelope or a cytoplasmic membrane, just a normal phospholipid bilayer sac, has embedded proteins. This is where you could find penicillin binding proteins. This is where you could find in gram-positives, lipoteichoic acids, which extend from this membrane to the outer exterior. So they'll extend from here all the way out through the peptidocell wall. These can induce tumor necrosis factor alpha and IL-1. And this is also the site of oxidative and transport enzymes. There can sometimes be, well, actually, do I have my, where'd my stain go? Okay, and maybe I discuss the stain later. We'll talk about gram-positive and gram-negative in a minute. Let me find it. So this would be a really good place. Okay. So we're going to just bump forward here. So gram stain, because I mentioned it, I do want to talk about it right now. So this is what gram-positive bacteria look like. It's very purple, gram-positive purple, P and P. Gram-negative bacteria appear red or pink, depending on what this color looks like for you. But bacteria that have thick peptidoglycan layers, which is your gram-positive bacteria, they're going to retain the crystal violet dye, which is the first dye that you use, better than bacteria that have a thin peptidoglycan layer, which is gram-negative bacteria. So when we do a gram stain, this has been tested on before, the order of a gram stain or dye is used, which is why I'm mentioning it. When you apply a gram stain, essentially you'll have bacteria that you've created a slide with. I don't know, anyone took micro in undergrad and had a micro lab? So some of you, so this will be very familiar. So you create your slide, you heat fix your slide, and then from there, you're gonna stain your slide. And so first step, you'll apply crystal violet stain. This is going to make all the bacteria appear purple, gram-positive and gram-negative. You then apply iodine to essentially fix the stain if you're a gram-positive bacteria. You then apply alcohol or acetyl alcohol or acetate, kind of depends on your lab. It's oftentimes like 95% ethanol. It's a pretty strong alcohol. You'll apply alcohol as a decolorizer. And what will happen when you apply alcohol to these stains is gram-positive bacteria, they have such a thick peptidoglycan wall that while some things may decolorize, meaning lose color, for the most part, they're going to retain the stain. They're going to retain that purple stain, that purple color. Gram-negative bacteria, they had a really thin peptidoglycan wall. So when you apply decolorizer, they're going to lose the purple stain, your gram-negative bacteria. So after this step, gram-negatives will look clear, gram-positives will still appear purple. And then at the end, you're going to counterstain with, there's a couple different counterstains you could use, safranin being one. And what will happen here is your gram-negative will now take up that stain because it's been decolorized and it'll appear pink or red. Your gram-positive will not appear pink or red because they already have retained the purple stain. They never lost the purple stain. And that's how you get your gram-positive and gram-negatives. So whenever I mention a gram-positive bacteria, it's looking purple. Whenever I'm mentioning a gram-negative bacteria, it's appearing pink. And the reason why is the layer, the peptidoglycan cell wall layer, its thickness, as well as how it's set up. We remember our gram-negatives, we go back to this picture here, our gram-negatives have an outer membrane, a thin peptidoglycan cell wall layer, and an inner membrane. Our gram-positives have this really thick peptidoglycan layer, but they do not have an outer membrane. So that's the difference between the two. Not every bacteria takes up stain, and we'll look at a few of the specialized stains or growth media that you may need to know for the test. But for the most part, they're going to test you on gram-positivity or negativity with these organisms. There's a few other structures that are kind of specialized that may be helpful to note. There can be bacteria that contain spores. This only occurs in gram-positive bacteria. So if you have a gram-negative bacteria, it cannot contain a spore. It does not have endospore forming properties. But bacteria that I have a slide that lists the common spore forming bacteria, but essentially it's a keratin-like coat. It has peptidoglycan, it contains DNA, and it helps these bacteria resist dehydration, heat, chemical damage, and or if there's not food or metabolic abilities for life. They kind of go into this suspended animation state where they aren't growing, they aren't dividing, they're not really alive, but they're not dead. And these spores can live for hundreds and thousands of years. And so these bacteria, because if they're endospore forming in healthcare settings, they can be harder to kill off if they are in an endospore state. And that's why we have things like autoclaves to try to destroy as many bacteria as possible, including ideally our endospore forming bacteria. So looking here, I don't know why I have urease just in the brand. Oh, and then I guess I don't know where my endospore went. I'm sorry, y'all. I feel like this is not all of my slides. Okay. I may have lost some slides. I'll look and see if I have a different version saved on my laptop on the break. Because the big spore formers I'd want you to know about would be your bacillus species and your Clostridium species. Those would be the two kind of big genus species categories I'd want you to know for endospore forming. I do have a slide somewhere on it. We'll see if we find it later. If not, I'll look up my slide set and see if I can post that updated one. One other component that's unique to some organisms is organisms that can produce urease. Urease can be helpful in keeping certain bacteria alive and living. And so these are your urease positive organisms, P chunks, because a lot of them are affecting or involved in the GU tract, right? Because that's where we're dealing with urea. That's where the urea cycle takes place. And so your Proteus species, your Streptococcus, H. pylori, ureaplasma, Nocardia, Klebsiella, and then your staph epidermidis and your staph saprophyticus, or strep epidermidis and staph saprophyticus. So these two at the end. So this would be your P chunks for urease positive organisms. Urease is going to be tested less than some other things like catalase, but still important to note. So these are a few stains and growth patterns that are helpful to know that could come up on NPLEX. Positive versus negative stain. Positive stains are basic stains. Bacteria typically are negatively charged thanks to their proteins, so they will take up a positive stain. So if you apply a positive stain, methylene blue being commonly the positive stain we apply, the bacteria themselves will take up the stain and they'll look like blue or purplish on your actual slide. Versus a negative stain, negative stains themselves are acidic. That means bacteria being negatively charged will repel the stain, so you can see morphology, the shape of the bacteria better, but you're not going to actually see the bacteria take up stain. The bacteria will be clear. The space around it will take up stain. So positive versus negative stain. The only time I see this tested on is when they're asking about potentially view under microscopy or morphology. They might ask, to view morphology, which stain would be more appropriate, and then they could potentially list some actual dye. Methylene blue is your most common positive stain dye. India ink is typically your most common negative stain dye, or nigrosin, Nigrosin, I believe. Those are your most common negative stains used. We talked about gram stain already. But not all bacteria gram stain, and that's important to note. So the bacteria that we talk about, I'm commonly going to say like these are gram positive, these are gram negative bacteria, but not all of them will take up stain. So this is your essentially little mnemonic to remember who doesn't. So these little microbes may unfortunately lack real color, but are everywhere. So these there's Treponema and Leptospira. These are too small or too thin of a membrane to take up stain. Microbes, these are cell walls that have high lipid content. Mycobacteria, Mycobacterium tuberculosis, right, are acid-fast bacteria, have too much fat in their, essentially their cell wall to take up the stain. Unfortunately, they don't have a cell wall. This is Mycoplasma and Ureaplasma, and then lack real color but are everywhere. These are microbes that are typically intracellular microbes. So Legionella, Rickettsia, Chlamydia, Bartonella, Anaplasma, and Uriclia. The most common ones that you'll see Treponema, that's causing syphilis. Mycobacteria, you'll see TB, Mycoplasma, walking pneumonia, and then Rickettsia and Chlamydia, obviously tick-borne illnesses, and then our common STI. These would probably be the most common ones you'd see from this list tested on. So what does an acid-fast stain look like? This is for your mycobacteria, so your tuberculosis-causing bacterium, and Nocardia. I included it here because it technically is the other one that does acid-fast stain well, but I doubt they would test you on it. But this uses carbofuscin and it turns bacteria red. So the red bacteria are the bacteria that are taking up an acid-fast stain. The blue are not. I have seen them use Zeal-Nielsen stain before on NPLEX. So acid-fast, Zeal-Nielsen, same stain, same bacteria, just different name. Acid-fast is used more conventionally in micro, but NPLEX seems to like calling things by the name for whatever reason. I'm not going to talk a bunch about agar. Agar is essentially the growth media that you're using to grow different microbes. Each microbe likes certain growth agar. TSA is like common growth medium or growth agar used, tryptic soy agar. But there are a few that sometimes NPLEX will pull because they're unique. Chocolate agar contains blood agar and then is heated up. And so it gets this chocolate or brown appearance. H flu, so Haemophilus influenza, stains very well on chocolate agar. And so I have seen this one come up before in the past. So that's why I have chocolate agar here. McConkey agar, this is essentially helpful with fermentation processes. It's going to tell you what types of sugar your bacteria like or don't like, specifically for lactose fermenting bacteria. These are commonly seen as your enteric gram-negative. Enteric meaning they like to live in the gut. So you can see this is an uninoculated plate, no bacteria. These are bacteria that are colorless versus bacteria that are pink. The colorless bacteria do not use lactose as a sugar or as a fuel source. So Salmonella, Shigella, and Proteus of your enteric gram-negatives versus these bacteria have created pink colonies, meaning they've used the lactose as fuel, which is E. coli, Citrobacter, Klebsiella, and Enterobacter. How they could ask a question would be on which agar would grow the bacteria associated with this case, or most likely associated with this case. And they may give you like a gastroenteritis case or some type of case asking about the enteric gram-negative bacteria. So then you're having to not only identify which bacteria is most likely associated with the clinical scenario, but which one would probably be a lactose fermenter or non-fermenter. If they're asking you about agar, they're probably going to want which one is actually going to show you the positive result. So you'd be wanting to know who are the lactose fermenters from this group. So these four here. Eosin methylene blue, EMB agar, E. coli creates this green stain. So E. coli with eosin methylene blue, E and E. And then this agar, Saborid agar, the only reason I have this here is because it's for fungi exclusively. It's too acidic for bacteria. So if there's any fungi question and they ask you about media, Saborid agar is your only choice. It is the option. So if it's non-bacteria, this is the agar that you're choosing. Those are the only agar I've ever seen tested on or asked on NPLEX. It doesn't mean they could pull others. There's hundreds of thousands that you could use and create, but these are the ones I've seen tested on in the past and their associations that they've pulled. So those are the only ones I'd focus on. A few components about metabolism. We're going to talk about anaerobes, aerobes, and facultative because I mentioned that yesterday in class. So your bacteria, some bacteria can only live in oxygenated states. That's much more rare. Most bacteria can live in either oxygenated or non-oxygenated states, meaning they're facultative. And then there are some bacteria that die from oxygen. They don't contain catalase. They don't contain essentially the enzymes to deal with reactive oxygen species. So they are only anaerobes. And we're going to look at the ones that are really more only anaerobes versus facultative here. So our only anaerobes, the anaerobes can't breathe fresh air. I mean, these will die off if they're exposed to oxygen states is Clostridium, Bacteroides, Fusobacterium, and Actinomyces israeli. Really, of this group, Clostridium would be the one to know, right? C. diff cannot live with oxygen. Hence why these are gonna cause infections in areas that aren't having an active supply of oxygen. So they're not going to be in your nasal tract, in your respiratory tract. They're not going to be on the surface of your skin. These are going to be microbes that are living in places where they can hide away from reactive oxygen species. Because if they're exposed to oxygen at all, they will not survive. Okay. When it comes to bacteria that like to hide in a cell, there's some bacteria that are obligate intracellular bacteria, meaning they cannot live outside of a cell. Why is this important? Because these bacteria then are going to only be found internally. They're only going to be able to be tagged by the immune system for things that can go intracellularly. And so the immune system is going to have to respond in a way kind of like it responds to a virus, right? That's always inside of a cell. So it's not going to be able to be necessarily targeted by some of our innate immune factors, our innate immune system that can go kind of find these bacteria around in the system, tag them, digest them, present them. These are inside the cell. So they take a longer for the immune system to identify. So our obligate intracellular bacteria that have to stay inside the cell is our little like mnemonic, stay inside cells when it is really chilly and cold. So it's Rickettsia, Chlamydia, and Coxiella. Okay, these are obligate intracellular bacteria, meaning they cannot live outside of the cell. They'll die. Facultative, they could live in a cell or out of a cell. And why this is important, because a lot of bacteria live outside of cells. They don't necessarily have to be inside of a cell. They'll actually live outside of a cell and they'll grow and divide. They're their own cells. They don't need to use our cellular machinery. But some bugs can use our cellular machinery or hide out in our cells as a way to protect themselves from our immune system. So these are some nasty bugs may live facultatively. And that's Salmonella, Neisseria, Brucella, Mycobacterium, Listeria, Francisella, Legionella, and Yersinia, our black plague. So of these to note, Salmonella, Neisseria, Mycobacterium, Listeria. These are more commonly tested on, but if you can remember the mnemonic, then you could remember them all. I have never seen Brucella or Francisella tested on before, so I would ignore those. Yeah. Correct, not aerobic. No, the only thing I would worry about when it comes to aerobes versus anaerobes is just know your anaerobes and then know pretty much everything else is going to be facultative. And then our last group is our catalase positive organisms. Catalase, again, kind of going back to the oxygen discussion, catalase degrades our H2O2 into water and oxygen. So this is a way that bacteria can survive essentially these reactive oxygen species. They can avoid being killed off by some of our normal processes of our immune system or our body. And so it's important to know who's catalase positive or negative. Of all the different funky tests and enzymes and things bacteria can have, catalase comes up the most on NPLEX because it's an relatively easy test. You just essentially put hydrogen peroxide on a little slide of bacteria, and if it bubbles, it's catalase positive. That's the micro test for it. It's very basic. So catalase positive organisms are big. Catalase, our cats have been two places. That's your mnemonic. So Bordetella, pertussis, H. pylori, Burkholdia, Nocardia, Pseudomonas, Listeria, Aspergillus, Candida, E. coli, Serratia, and Staphylococci. So these are the catalase positive organisms. Of note here, staph species are catalase positive. When it comes to other gram-positive cocci organisms, we're going to look at staph and strep together. A big separator is staph is catalase positive, strep is catalase negative, typically. So it's a way that you can separate these really, really closely related bacteria. Okay. That was our first little basic intro. We're going to look now at our gram-positive and gram-negative cocci. So that's going to be what we're going to look at next. Like I said at the beginning, we're going to really focus on bacteria today and then go a little into viruses. We're going to pretty much ignore the other microorganisms. They're not high yield at this point for the test. Okay, so these are all of our gram-positive or gram-negative cocci shape, so circular shaped bacteria to note. So that's what we're going to go through. We're going to mainly pay attention to staph and strep initially, and then we'll talk about some other cocci morphology bacterium that are important to understand. So Staphylococci, so this is a genus of bacterium. We're going to look at multiple species. Some general characteristics, they often live in the skin and mucous membranes. They are spherical, so they're circular in shape, and they're arranged in irregular clusters. This is where you get the clusters of grapes appearance. They are gram-positive, they do not form spores, and they are non-motile. They may or may not have a capsule, depends on the species. There's 31 species to note. We're not going to talk about all those species. We're only talking about a few of the important ones. If you're a table type person, This is going through all of the pathogenic gram-positive and gram-negative cocci. So all of them in the whole lecture. It'll give you the organism. It'll give you some places you could find it in the skin or what it could cause in skin skeletal, if it causes a nervous or a muscle condition, cardiovascular, GI, respiratory, urogenital. So this can be a great place to start if you're really trying to match like path to disease or path to bug. And that can be a good place to start really getting some name recognition from these organisms, is identifying what bug causes what condition or what disease most commonly. Something of note is you can see Staphylococcus aureus is commonly seen in things like cellulitis, in boils and carbuncles and abscesses, but Strep pyogenes is common cause of erysipelas, which also can look like cellulitis, but typically is more seen on the face. So these are two different skin, red, irritated, indurated conditions caused by two different organisms, both gram-positive, both cocci-shaped, two different genus. So when I have something that's that close, that's a good separation. Staph aureus, cellulitis, Strep pyogenes, erysipelas. So that's how I would use kind of a table like this to try to separate out these bacteria with their diseases that they cause. This is again listing more of our pathogenic gram-positive and gram-negative cocci in table form, showing you what conditions they are associated with. We're going to go into each of these bugs closer. So back to our staph species. Our first staph species I want to talk about is Staph aureus. If you know one staph species, know Staph aureus. Okay. It grows in large, round, opaque colonies. It's facultative anaerobe, so it can deal with both oxygen or non-oxygen states. It can live in a lot of different kind of extremes, so high salt, high pH, high temp, and it has lots of ways to infect someone, lots of virulence factors. These are a list of virulence factors of Staph aureus. Don't know all of them. Don't memorize this list. Understand that it does a lot. When in doubt, Staph aureus probably does it when it comes to a virulence factor. Okay, it's not true for everything. As you can see, it has a lot of abilities, Staph aureus. These are its enzymes, these are its toxins. A couple interesting things that Staph aureus can do is it has coagulase, which is an enzyme that can coagulate our blood plasma. So this can cause clotting potentially. It also has penicillinase, which can inactivate penicillin. So this is how we get some bacterial resistance. And then another interesting component is some of its toxins down here, specifically toxic shock syndrome toxin. This is literally the toxin that we see in toxic shock syndrome, hence its name. So fever, vomiting, rash, organ damage. So a lot of these toxins can really cause some severe pathology in the body. Staph aureus is present in majority of environments that are frequented by humans. We can see it in fomites, so on surfaces of things. There's a pretty common carriage rate for people. Most people carry it as normal healthy carriers on the skin, in the nares, in the nasopharynx area, or even in the gut. If you're more likely to get this, so risk factors of a Staph aureus infection, poor hygiene states, malnutrition, tissue injury, immunodeficiency, this is common across the board, right? So for all of these bugs, we're going to see very similar reasons why you could get infected. And this is the bug that's associated with MRSA resistance, methicillin resistance. So that is one important component to note. You can have local infection with Staph aureus or systemic. So localized could look like a folliculitis, a carbuncle, or furuncle, and impetigo. All of these skin conditions, Staph aureus can be one of the major players, if not the major player. Knowing the difference between what is happening, so folliculitis, inflammation of the follicle, a furuncle or carbuncle, these are those abscess-like shaped structures that have purulent discharge in them. Or impetigo, these are our bubble-like swellings, often more seen in newborns that can break and kind of peel away. So lots of different skin conditions with Staph aureus. You can also see things that get deeper. So osteomyelitis, if osteomyelitis is on there, Staph aureus would probably be my #1 choice for that condition, for the bug that causes it. Inflammation and infection of the actual osteum, the bone, or bacteremia. You can see sepsis or full body infection. And we think about that when it comes to endocarditis, right? Staph aureus being the most common cause of endocarditis in someone that has healthy valve tissue, healthy heart tissue. You can see toxigenic disease. These are diseases caused by Staph aureus's toxins that we saw. So food intoxications, this is where you get your gastroenteritis or your food poisoning, staph scalded skin syndrome, or toxic shock syndrome. So these are staph diseases thanks to staph's toxins as their virulence factors. All right, so that's Staph aureus. When in doubt, if you have a staph question, choose Staph aureus, like the most common one you'll see. There are some other staph, though, bacterium that are important to know. These are all coagulase negative staph. So Staph aureus is the only coagulase positive staph species. Coagulase negative staph account for a large proportion of healthcare associated diseases or opportunistic infections, especially in immunocompromised patients. So staph epidermidis is probably your next most common. It lives on your skin, lives in mucous membranes. You could see this in endocarditis, in sepsis, in UTIs, less common. Staph hominis, I would not worry about this. Staph capitis, scalp face, external ear, so some external infections. All three could cause a wound infection by penetrating the skin. And then Staph saprophyticus doesn't live on the skin, likes more the intestine, vagina, and can cause UTIs. So of these, I would just note that these are coagulase negative staph species, more associated with healthcare associated or immunocompromised associated infections. Okay. If you're like wanting to go through the microbiology of the all, I do have some like flow charts that you can kind of try to separate out via identification, these species. I would say for the purposes of the test at this point, I wouldn't worry about this, but if you're really trying to understand, like how would I go through and differentiate these species from one another, this could be a way that you do it. Essentially, it shows like anaerobic growth, urease production, ferments mannose, staph epidermidis, does not ferment mannose, staph saprophyticus. It's essentially walking you through a dichotomous key. Dichotomous key is used to kind of figure out what bacteria did it from just a basic sample of an unknown. So at the purposes of this time, I wouldn't go through these weeds, but if you really are trying to differentiate, this might be helpful in your future practice, to be honest, helping you figure out what tests you could order. Yeah. Coagulase is going to coagulate blood plasma, and it's only seen in Staph aureus. That's think more strep species for your alpha, beta, gamma hemolysis, but it does have a component of beta hemolysis, and it's not necessarily due to the coagulase. It's due to one of its toxins. It has our staph species. If we look back at its toxins list, we can see that it has hemolysis alpha, beta, gamma, delta, and that's how it's lysing red blood cells, but that's its toxin. Its coagulase component, it actually can create clots and coagulate blood plasma to essentially avoid immune cells coming in. So it can kind of help itself wall off its own little infectious region. It's how it can be so good at creating vegetations on valves. It's why it can wall itself off and create an abscess in the skin. So it's what makes it unique and able to essentially isolate itself from the immune system, versus the lysing of the cells is due to this toxin, the hemolysis toxin. Yeah. Good clarification. Awesome. The one big other clinical concern, I already mentioned this, is the MRSA, the methicillin-resistant Staph aureus. 95% of staph infections have penicillinase and are resistant to penicillin and ampicillin. So hospital-resistant or antibiotic-resistant infections of Staph aureus are common. This is not a pharm class, so you likely won't be asked about any pharmacology whatsoever, but penicillinase, MRSA, those are fair game on this test. So if there is a question about a patient was given penicillin for an infection and their infection got worse, then your bug could have a penicillinase ability to bind penicillin, a penicillin binding protein, and essentially render it inactive. And so think about staph species as one of those common bugs that have penicillinases, the ability to bind penicillin and render it inactive. MRSA obviously has resistance as well to methicillin strains, and often multiple antibiotic resistance. So it's trickier to treat. And typically you have to like lance abscesses, debride things, you're going to do multiple things to treat these infections. Okay. All right. So of all the tests, which tests differentiate Staph aureus from other staph species? Coagulase, yeah, coagulase. If you already said, sorry, I couldn't hear you. I heard a lot of like, I was like, okay, I don't know. Coagulase, yes, coagulase is the only positive in Staph aureus. So coagulase will not be positive in any of the other Staph species, only Staph aureus. So that's what differentiates Staph aureus from all the other Staph species, right? There's other things that will differentiate the genus of Staph from other geni, right? Or genus of other bacteria. But Staph aureus, coagulase. Ooh, look at me. It's 1 a.m. Okay. Strep species. Our other gram-positive cocci that you should know is our strep species, Streptococcus, Streptococci. So some general characteristics. These are also gram-positive. They're also spherical, so circle in morphology. They, instead of in clusters of grapes, they're in long chains. Typically, that's how they're arranged. They can sometimes be in pairs, diplococci. They don't have spores. They're also non-motile. They can form capsules or biofilms via slime. They're also facultative anaerobes. We're seeing a lot of comparison and crossover between Staph and Strep. They do not form catalase, so that's a difference. Staph, catalase positive, Strep, catalase negative, but they do have a peroxidase system, so they can still deal with oxygen, but they don't deal with it via catalase, okay? Most parasitic forms, meaning most forms that cause pathology or disease, are fastidious. It means that they need certain media to grow. They are, whereas Staph maybe eats anything or consumes anything, Strep is a little bit more picky about what it likes to eat and consume for optimal growth conditions. Their colonies are small and whatever, it's fine. Okay, so when we look at Strep subgroups and break them down, we break them down often based off their ability to do hemolysis. And so this is kind of what Ali was already getting us into the conversation is who's doing what type of hemolysis and what does that mean? When we're talking about this, often we use this as a way to differentiate the species on growth media. So that's where it's commonly discussed and talked about. We can also look at like the pathology or the physiology behind the hemolysis. For the actual purposes of NPLEX, they're not going to ask you, how does alpha hemolysis work? How does beta hemolysis work? They are going to have you want to know who are beta hemolytic versus alpha hemolytic strep species. They aren't going to ask you the whys about it. They're not going to ask you what a media looks like. They're not going to ask you about what's actually happening physiologically. So memorization here would be probably your best bet when it comes to these organisms. So our beta hemolytic strep species are going to be our group A strep, which is strep pyogenes, and group B and C strep. We'll look at these a little bit further here in a moment. Our alpha hemolytic group is going to be strep pneumoniae, and our group D strep, as well as viridans, strep viridans. So these are going to be our alpha hemolytic organisms. Now, when we look at these, we can see here, if this was not good for your view, this is showing the exact same thing: beta hemolytic, group A, group B, alpha hemolytic, strep pneumoniae, and then group D or viridans. I wouldn't necessarily worry about the antibiotic resistance, although we did see a test question that talked about optogen, right? So could they throw those at you? Sure. But remember, we didn't need to know if that bacteria was optogen resistant or sensitive to actually get the correct answer. As long as you were able to associate the pathology, you could get there. So you don't often have to have that in-depth micro-knowledge to understand these questions. This is kind of an easier picture for some. It goes through and shows you the species of strep, and it shows you the hemolysis type. Lancefield groupings are another way that they've grouped or organized these organisms. I've never seen Lancefield groupings asked on NPLEX, so you could ignore this category. I have seen hemolysis and then habitat or pathogenicity. Obviously, those are important. Strep pyogenes, obviously a common strep species to be tested on. Strep agalactiae, maybe, but definitely strep pyogenes. Know that it's beta hemolytic, and this is your classic strep pharyngitis, so human throat. And then if left to its own devices, could cause scarlet fever, right, as a complication. Some other strep species that would be important to know: strep mutans, strep pneumoniae. These would be helpful ones to kind of have in your back pocket. Strep mutans commonly seen with dental caries. Strep pneumoniae, it's alpha hemolytic respiratory tract. We're thinking about pneumonia, right? So these associations can be helpful here. I have the species in bold are the most significant sources of human infection and disease. If there's a N, it means none. So hemolysis none for strep mutans. If it's a V, it means it could do probably multiple types of hemolysis. So we look at first at our beta hemolytic strep pyogenes. This is the most serious pathogen of the strep species or strep genus. It's strictly a parasite, meaning it's only going to cause pathology. That's what I mean when I say parasite, okay? It also is going to need to live within us to cause that pathology, right? So we're not going to see it infecting a fomite and causing, it's going to have to infect us to be pathogenistic. When it inhabits us, it's going to inhabit our throat, nasopharynx, and potentially the skin, but really think throat, nasopharynx as your two big areas. What can it do to us? These are some of its virulence factors. It has multiple different surface antigens. Some important ones are listed here. It can protect itself against lysozymes, so it can protect itself against some phagocytosis. Fimbriae can adhere to things. That's why it's really good at sticking to our tonsils, right? It can adhere and actually create this exudate that you can see in a strep infection. Another protein that can prevent phagocytosis. By having a capsule, it can provoke no immune response. So it has to get opsonized to be able to be phagocytosed. And it can also inhibit some of our innate immunity via complement and neutrophils. So it's really good at creating an infection, essentially. Some other virulence factors of strep pyogenes, we have extracellular toxins and extracellular enzymes. So some extracellular toxins, streptolysins, a pyrogenic toxin with fever, superantigens, all of these are going to cause, essentially like the staph toxins, they're going to cause more symptoms, more pathology in humans. And then we have our enzymes. These can digest things, right? So we can break down fibrin clots, we can break down connective tissue, we can even break down DNA. So these are some of its enzymes that it can secrete and use to its advantage. So the only reservoir for strep pyogenes is humans. We can also carry it. Transmissions, typically droplets. You could get it from fomites, food contact. Typically enters via the skin or pharynx. And children are mainly affected for the cutaneous and throat infections. You can, though, see, as we've seen in other lectures, systemic infections and potential sequelae for untreated strep pyogenes infection. Impetigo is one that you can see topically. So Staph aureus and strep pyogenes can cause an impetigo. You'll see these more associated with insect bites, poor hygiene, crowded living conditions. Both can occur, though. So these would be either or would be a good answer when it comes to an impetigo question. I would probably choose Staph aureus over strep pyogenes if I had to choose between the two, but both are good answers. Erysipelas, this is specific for strep. So if I get an erysipelas question, I'm choosing strep pyogenes every time. The pathogen enters into broken skin. It gets into the dermis and sub-Q tissue. This type of cell, cellulitis, or I guess skin infection, is different in cellulitis in that it's raised. You can actually see the dermis will have a raised layer. Compared to cellulitis, you'll feel the skin will be hot, it'll be hard, it'll be painful, but it'll feel flush. There's not going to be a raised area. Erysipelas, though, you'll feel the skin and you'll actually go up like onto a platform over the area that's infected with erysipelas. And I'm touching the face because it's more commonly seen in the face, but you can have erysipelas anywhere, but typically it's more likely an infection of the face. Often the ear is where it starts. The tissue is more susceptible. Strep pharyngitis, that's the one we think of, a classic strep pyogenes infection, strep throat, and then potentially a sequelae, scarlet fever. This kind of strain of strep pyogenes has the erythrogenic toxin that causes that fever, that redness that we classify with scarlet fever. This can lead to, obviously, full body infection. You can see with strep pyogenes a pneumonia or strep toxic shock syndrome. Pneumonia, though, we often associate more with strep pneumoniae. Complications of group A infections, rheumatic fever or acute glomerulonephritis. So your strep pyogenes that's really your condition, your bacteria that could lead you to a rheumatic fever or an acute glomerulonephritis condition. And that obviously severe sequelae commonly tested on in association with these bacteria questions. What's the bacteria associated? What condition could they have had that led them to this? Or what's the complication? So recognize, kind of put these in your brain, lump them together. So what's not a possible complication of strep throat? Pneumonia, exactly. While you can have a strep pyogenes pneumonia, right, it's not a complication from strep throat. That would actually be its own separate infection versus strep throat, strep pharyngitis could lead to rheumatic fever, scarlet fever, or acute glomerulonephritis as complications. Yeah. Yeah.
It's just a term that you use to say as rattles, like red throat, fever, and
no, it's a separate condition than strep pharyngitis. Yes. Correct. Yep. So scarlet fever, think scarlet fever, its own condition, rheumatic fever, its own condition, strep pharyngitis, its own condition. Strep pharyngitis associated with our Centaur criteria, the erythematous throat, painful throat, fever, typically under the age of 12, no cough, and exudate on the tonsils. But typically you're not seeing an external body rash and you're not seeing a red cherry tongue. So there's some differences from strep pharyngitis to then scarlet fever. Scarlet fever, you're starting to see the erythrogenic toxin being displayed, which gives you the bright red rash, bright red tongue, bright red mouth. Whereas with strep pharyngitis, you might just see a red throat, typically. Yeah. And then with rheumatic fever, we have all those criteria. So that's kind of how I would scale them out. Any of them can be severe or serious on their own. If I had to order them in severity, I'd say strep pharyngitis, least severe, scarlet fever, maybe next, and then rheumatic fever, the worst. Yeah, excellent. Great. So like Staph aureus, if we have to know one strep species, no strep pyogenes, okay? Obviously there's more, but strep pyogenes would be your most important one to know of the strep species. Another strep species that is secondarily important, if you can learn two in your brain, is strep Agalactiae, which is our group B strep. Okay, so group A strep, strep pyogenes, beta hemolytic. Now we're dealing with group B strep, that's strep Agalactiae. We see this more common in the human vagina, pharynx, and large intestine. It can be transferred to infant via delivery because it can colonize the vagina. And so this is the most prevalent cause of neonatal pneumonia, neonatal sepsis, and neonatal meningitis. So those are kickers. They love asking you, like, what is the most common aged person to get this type of meningitis infection, right? So neonates, pneumonia, sepsis, meningitis, think group B strep, strep Agalactiae. So this is why pregnant women are screened and treated if they are infected with group B strep. Wound or skin infections you could see with this, but less common, as well as endocarditis in someone who's immunocompromised. Sorry. There we go. All right. We also then have group D, group C, group G strep, all these groups, right? So group D is actually not called a strep at all. It's Enterococcus facilis or facium or durians, okay? But this is still categorized under the strep species, under the strep umbrella. This is a normal colonist of the large intestine. So this would be part of normal healthy flora, but it can opportunistically cause urinary wound or skin infections in someone who's debilitated or immunocompromised, okay? Group C and D strep, it's frequently isolated in upper respiratory infection, like upper respiratory flora, so it could be normal flora. Again, in someone who's immunocompromised, we could see a pharyngitis, a glomerulonephritis, or bacteremia. I wouldn't worry about that one as much. Okay. This is the schema that I gave for staph, just for strep, specifically beta hemolytic streps. And I don't really care about treatment. Okay. All right. So now we're moving from our pyogenes, there's our group A, our Agalactiae, our group B. Now we're moving out of beta hemolysis and into alpha hemolysis. And we're talking about our alpha hemolytic strep group, the viridans group. That's the first one we're going to talk about. So strep viridans. This is the largest complex group. It has all these different subspecies that are included in viridans. So recognize when we say strep viridans, we're actually including 6 different species of strep within that title, that name. They're probably not going to break out these individual names for you. The only one I've seen them do, and out of strep viridans, often they'll just put strep viridans. But if they do separate it out, strep mutans is the one I see. And so that's why I do have that one listed out in full. So if you see strep mutans, it is in this viridans group. Okay, so you can assume those things are true about it. They are the residents of the gums and the teeth, the oral cavity. So this is our dental associated infection. This is where we think about getting an endocarditis after getting a dental work done for someone who has valve disease, right? That's our strep viridans group. Typically they stay in our mouth. They're not invasive unless if you have work done, right? Unless if you have a procedure or something that's completed. So the viridans group, again, if it does cause an infection, it can cause things like bacteremia, meningitis, an abdominal infection, or even an abscess. But the most serious infection, and the only one I care that you associate, is subacute endocarditis. You get this bloodborne, it settles on heart lining or valves, specifically people who have pre-existing heart disease or at high risk. It is really good at forming biofilms. So that is why it can be hard to detect and it can be really damaging to the valves. So where I said Staph aureus, you're going to know for acute endocarditis, strep viridans, you're going to know for subacute endocarditis, and subacute endocarditis is the kind that's also associated with damaged valves or some type of heart condition. Okay. All right. I already said it can create slime. It can create a biofilm. Okay, and then the last one that would be the most important would be our strep pneumoniae, and then we'll break. So strep pneumoniae, this causes the majority of our bacterial pneumonias. So if you had a question, what's the most common cause of bacterial pneumonia? Strep pneumoniae would be your best bet. It doesn't have catalase or peroxidases. Cultures will die in oxygen, which is ironic since it's in the respiratory tract. Let's see if there's anything that's helpful here. Nope, pretty standard. I'm seeing if there's anything else I would care that you know. No. Okay, great, excellent. So that's our gram-positive cocci that I want you to know. Know your staph species, know Staph aureus in particular. Know your strep species, Strep pyogenes, Strep agalactiae, and Strep viridans, and Strep pneumonia. So 4 strep species to 1 staph species. Those 5 bacteria will serve you for a good majority of your micro questions if you're able to associate some general virulence factors and the pathologies that they're associated with. So we'll take a 10-minute break. I'll look at the questions on the chat while we do. We'll come back at 10 after. We'll finish up our cocci and we'll talk about our bacilli bacteria in this next hour. So 10 after, we'll get started again.